National 5 biology students. Let's do these past paper questions together. So number 5 here says, which diagram shows the concentration of product during a degradation reaction? Now, when a degradation reaction is happening, we are breaking down a large molecule into smaller molecules, okay? And so therefore, the concentration of the product is going to be high as the reaction occurs. So as time passes and more of this reaction occurs, the product that's produced is going to become greater and greater, okay? And because of that, our answer here has to be C, okay? Because the product is continually being produced, so its concentration will go up and up and up and up and up, okay? If the question was, what's the concentration of the substrate, then B would be our answer here, okay? But because it's the product, it's going to be C there. Number 6, the following stages occur during the process of genetic engineering. And okay, which of the following stages shows the order in which they occur, right? So first of all, we need to identify the chromosome, okay? That isn't one of our options. Second of all, we have to remove or extract that gene from the chromosome, okay? So that's going to be that one there. That'll be number 1, sorry. And then we need to get the plasmid from the bacterial cell, so that's going to be number 2. We then need to cut open the plasmid, number 3, and then we're going to insert the gene into the plasmid, so that's going to be in that order there, okay? So it's going to go 4 to 3 to 2 to 1. So that's going to be D there as our option, okay? And if you don't know that one, you need to memorise the steps involved in genetic engineering, okay? You need to memorise those. Number 7. A group of scientists investigated the effect of temperature on the growth of genetically modified cells. The GM cells were grown in nutrient solution at 20 degrees. This was repeated at 30 and 40 degrees using fresh nutrient solutions at each temperature. Which of the following would improve the reliability of the results, okay? So as I always say, remember re, okay? When you want to improve the reliability of the results, you need to repeat what you're doing, okay? Repeat the experiment or the investigation. Now, let's have a look at the options we have here. They set up the nutrient solutions containing GM cells at 25 and 35 degrees. Well, that's not going to improve the reliability because these are different temperatures to what we had originally, so that's no good. Set up 10 nutrient solutions containing GM cells at each temperature. Now, if we set up 10 solutions, that sounds to me like we're repeating it, we're doing the same thing 10 times. Because we're doing it at each of the temperatures. So I think that's going to be the right answer, but we'll come back to it. C, use the same concentration of nutrient solution at each temperature. Well, that is good practice, that's good to do in an experiment, but that would affect the validity of the results. That would improve the validity of the results, okay? Not the reliability, so that's why we're not using that one. And using the same types of GM cell at each temperature, again, would improve the validity of the results, but not the reliability. Okay, so that's going to be B for that one there. Okay, folks, hope that's helped. I'll see you in the next one.